Okay, so welcome back, everybody, for our next talk today of Kazuyoshi Ishigaki. We'll talk about HLA autoimmune risk alleles and how they restrict TCR CD3s. Please, Kazuyoshi.
Okay, can you hear me?
Yeah, we hear you and we see the screen. And
Okay, great. Hi everyone. I'm Kazuyoshi Ishigaki from RIKEN Institute in Japan. I'd like to present a talk titled HLA autoimmune risk restrict TCR CD3. This is a unique type of genetic research on TCR CD3 amino acid composition. Okay, let me start. So first, I'd like to start my talk explaining how I came up with this research idea using sort of my career path. So my background is medical doctor. I was trained as a clinical immunologist, and my specialty is rheumatoid arthritis. And rheumatoid arthritis is an autoimmune disease which immune system destroy joint, and the patient suffering from this joint disabilities. And during my PhD training, I conducted TCR study from sample from RA patients. And after that, I tried to change my research areas, and I studied human genetics. And after I finished some of the postdoc period, I was highly motivated to connect all of these my specialties, and I end up studying to do conduct RA genetics, which study trying to connect RA genetics and TCR CD3. And during my postdoctoral training at Harvard University, Harvard Medical School, and the result is reported just recently to Nature Genetics, and this is going to be the main topic of this talk. Now I moved to back to Japan, and I started my own lab in RIKEN. So I think many of you are familiar with TCR, but I don't think many of you are familiar with genetic. Just to make sure everyone on the same page, I prepared this, right? So genetics is a study genomic variants in our genome. And in the genome, we have millions of variants, and some of the variants increase the risk of rheumatoid arthritis, and some of the variants decrease the risk of rheumatoid arthritis. And genome-wide association study, GWAS, have identified many risk variants for rheumatoid arthritis, and we have currently identified many risk variants for RA. So what are they doing? So what's the mechanism of RA risk variants? And this is my research question for years. And this is this figure is showing the summary result of RA GWAS, and y-axis is the minus log 10 p-value, so which means the significance of association, and the x-axis is the chromosomal positions. And as you can see here, one of the striking genetic signal in our genetic study is signals coming from HLA genes, and actually this is accounts for about 50% of genetic risk of RA, and again, this is going to be the main topic of this talk. And the rest of 50% is explained by non-coding genome-wide non-coding variants, and going to be affect the cytokine pathway with transcription binding. And but this is not the main topic of this talk. So so what the mechanism of the HLA, I mean HLA risk alleles in our genetic studies. This is a key question of this my research, and the very important question to think about to answer this question is where the specific position of the risk allele. And the previous study demonstrated that these substantial strong signals can be explained on only a few amino acid allele in HLA DRB1, and especially position 13 and position 71 of HLDRB1 gene have confer a lot of genetic risk of rheumatoid arthritis. So please remember position 13 and 71 of HLDRB1 because I come back to this position later for coming slides. So this is a schema of how HLA is interacting with TCR, and I think many of you know that HLA present antigen to TCR, and this amino acid of RA positions around here at around the center of antigen binding pocket, and so it physically interacts with antigen, and the antigen are recognized by TCR CDR3 complementary determining region 3. So basically the HL risk amino acid can be interact with CDR3. So just the super basic background. So that basically two random recombination process produce highly diverse TCR, and so basically those junction region is a CDR3, and as you know, the center of CDR3 has super high diversity, and so basically there are, broadly speaking, two categories of the scenario where HLA risk arrow has some influence on the immune system. And the one very popular, well-studied hypothesis is peripheral hypothesis, where the HLA risk amino acid have higher affinity to pathogenic epitopes, like stenotypic epitopes in RA, and that because due to this high affinity, high efficiency present key epitopes, the HLA risk allele confer increase the risk of developing disease, but at the same time, we need to think about this hypothesis, named central hypothesis. So basically, HLA risk amino acid can influence a T cell selection in thymus and can select some specific TCR which will induce autoimmunity. And actually, this hypothesis is not well studied, especially in terms of genetics. So we decided to ask this whether this central hypothesis is reasonable in genetics association. So we what we conducted is to test association between HLA genotypes and TCR CDR3 amino acid composition, and we named it the CDR3 QTL. QT is a type of study, type of genetic association study. So basically, CDR, actually, this is the first formal comprehensive genetic research asking the association between those two factors. So in this study, I used two datasets. One is Emerson et al. Dataset published in Nature Genetics. I used this as a discovery dataset. And so Emerson et al. Dataset is published in Nature Genetics a while ago. And I think this is a relatively famous paper, and some of you might know this, but basically in this paper, they created a huge dataset which have containing TCR CD3 sequencing information plus HR genotype data from around 700 donors, and all of the data are publicly available. And this data is generated by adaptive biotechnologies. And in this Immunosec web portal, we can download all of the key dataset. So obviously, the TCR primary information of TCR sequencing is, of course, a bunch of CD3 sequence data. And actually, the sequence data itself is a bit difficult to handle in genetic association study. So we need to do something to do genetic tests. So this is my strategy. It's very simple. So basically, when I focus on one specific position, let's say position 110 of CD3, and when I want to ask the allele in this example, I converted this sequence into simple amino acid ratio data. For example, in this individual A, the alanine usage at this position is 3 out of 9, and this for this individual 6 out of 9. So using this type of simple calculation, we can convert sequence data into simple quantitative data. So after converting the sequence data to quantitative data, we can easily ask the association between TCR CD3 amino acid composition versus HLA amino acid allele. And basically, in this study, I use 2 different statistical model. So basically, in this my association test, I focus on one specific position of TCR CD3 and one specific position of HLA amino acid. And in one position of TCR, we have up to 20 different amino acid. And in HLA side, if the position is very highly polymorphic, then it has up to 6 different allele. And in one model, I asked the correlation between those two using all of the amino acid possible combinations. And I used MMLM, multiple linear regression model. And in the other model, I used a super simple strategy. I asked each amino acid at one time using simple linear regression model. So this is one of the highlights of my research result. So here, y-axis is the association minus -10 p-value of association, and the x-axis is the HLA amino acid position. So this is a HLA amino acid wide study, and so the top hit, the lead association was observed in position 13 of HLA-DRB1, and the p-value is super significant, less than minus 10 to the negative 100, so super significant. After controlling the position 13, the second hit was observed in HLA position 71 of HLA-DRB1. Those are super exciting results for us, because those position 13 and position 71 of HLA-DRB1 is identical position as the association we observed in our genetic study. So basically, those findings indicate that HLA risk of RA may be mediated by TCR CD3 amino acid modifications. And so just showing the association is not that informative. So we dive into deeper in this, and we ask more specific questions. And by the way, when we observe super significant p-value, we need to confirm that p-value is well calibrated. In other words, the type 1 error rate is well controlled. And so we conducted permutation test, and I'm showing here permutation p-value, and as you can see, there's no significant association at all. So basically, the p-value, super significant p-value in the observed dataset is real, no weird inflation, so which is good. And this heat map is showing the same association, but focusing on different metrics. This heat map showing the variance explain the association strengths, and in this heat map, y-axis is the HLA DRBM amino acid position, and x-axis the CDR3 amino acid position, and red color indicate the very high variance explained. So as I said, the position 13 of HADRBN has the strongest association, and another important information that the strongest association was observed at the center, very center of CDR3, not the peripheral region of CDR3. So, as I said, the center of CDR3 has super high diversity. So, it's a bit interesting result for us, because the center has basically the very random the amino acid which are created by very random process actually has more restricted by HLA genotype. This is the key message of this panel. So this next we try to replicate those associations observed in emasculated dataset, and we used in this replication we use RNA-seq data from naive T cell, naive CD4 T cell published in Blueprint Consortium. Although RNA-seq is of course not indicated to test TCR, but we got sufficient amount of T cell sequence reads from standard RNA-seq data. And so here I'm showing you the effect size comparison between the discovery dataset, Emerson et al. Dataset, and replication dataset. And we observed super nice replication result. And so we were very confident about our association test. And here we asked more specific questions. So here I'm plotting the effect size of our association test between the effect size in array GWAS. And here is the association to the in this is the association at HLDRB on position 13, and position 13 have 6 possible amino acid showing in each one plot. And this is the effect size for aspartate acid in position 110 of CDR3. So here you can see a nice positive correlation. So it indicating that the risk of array increase this amino acid. And in a reverse way, when we look at the lesion at the same position of CDR3, we observe the complete opposite trends. So the risk array risk decrease lesion usage at this position. And aspartate is a negatively charged amino acid, and lysine is positively charged amino acid. So basically, those are a result indicate that array risk of HLA amino acid at HADR site 13 decrease CDR3 positivity. So till now I'm focusing on HLA position 13, but here I try to extend the HLA risk amino acid. So as I said, 71 and other position is also important in RA or other autoimmune diseases. And so I calculated HLA-wide genetic risk for RA, and I named that this score as a HLA risk score, and I conducted CDR3 QT analysis using HLA risk score. So the result are summarized here. So This here figure indicate that the positive effect size in this figure indicate that HL risk, total HL risk of rheumatoid arthritis increase the amino acids at this position of CDS3. So this is consistent with the previous finding, the HL risk of rheumatoid arthritis increase these acidic, which is negatively charged amino acid, but decrease positively charged amino acid. And we also conducted almost similar analysis to type 1 diabetes and celiac disease. And for type 1 diabetes, we observed similar things. The risk, genetic risk increase acidic, decrease the basic amino acid. But in celiac disease, we observe slightly different trends. So genetic risk of celiac disease increase hydrophobic amino acid in CDS3. So those CDS3 modification associated with HL risk maybe increase the autoreactivity to pathogenic epitopes. So the short summary to here that we show that HLA risk amino acid strongly influence T-cell CD3, and there seems to be disease-specific patterns like this, and next we wanted to ask whether this pattern is really pathogenic to autoimmunity. So in order to ask this question, we created a scoring system named CDS risk score. This risk score is can be calculated for each CDS3 sequence, and if given CDS3 sequence have enriched for these risk CDS3 pattern, then the score will be high, and if the opposite is true. So if those CDS3 TCR T cell which have this type of TCR is really pathogenic to RA, then the RA clinical sample, the T cell which is specific to pathogenic epitopes in RA patient should have higher CDS risk score. So we tested that using the clinical sample. So we applied the CDS risk score system to RA clinical sample, and we collaborated with the researcher in Villarreal Institute, and we calculated CDS risk score of T cells which are specific to sterinated epitopes, RA pathogenic epitopes. Although number of TCR is very few, we can see that the T cell which is reacting to sterinated epitopes in RA patient synovium have showed a higher CDS risk score like this. So basically this is a positive result supporting our hypothesis. So in summary, we show that we wanted to provide genetic evidence supporting central hypothesis, and we demonstrated that HL risk allele of autoimmunity strongly influence amino acid CDS3 amino acid composition, and we found disease-specific patterns like this. And the CDS3 score analysis showed that some of those features were enriched among T cells specific to candidate pathogenic antigens. So this is a summary of the finding, and yeah, this is acknowledgement. So all of the analysis were conducted during my postdoctoral training period in Harvard Medical School, and at the Ray Chowdhury lab, as the Shoma Ray Chowdhury is the PI, and I had a very nice, many nice discussion and input from him. And I also like to thank the key collaborators in the Ray Chowdhury lab and outside the Harvard. Yep, that's it. And yep, thanks for listening. I'd be happy to take any questions.
Thank you very much. Thank you very much, Kazuyoshi. It was very interesting. Well, I'll let first the audience ask questions, and then I have several of my own. So any questions? Okay, while people are thinking, I can start. I mean, at the beginning with the kind of Manhattan plot, I mean, there were other locations as well that had relatively low p-values. Not here.
Yes,
I mean,
yes.
first one, in particular, I remember one in chromosome 1. I mean, did you explore?
You mean the?
Yeah, I think it was here. Yeah.
Okay. You're talking about this association?
Yes, yes.
Yeah, this is a association coming from PTP and 22 missense variant. So basically, which is a very famous risk allele for RA, and also actually for other autoimmune diseases as well. So the PTP and 22 also there are several hypotheses, but maybe the TCL downstream signal will be modified by these variants. So combination of HLA and PTP2 association suggests that TCRs, MSC TCR signaling is a key, signaling in the biology pathology of RA.
Okay, thank you. I see that Corey Watson posted a question in the chat. How does your model incorporate inputs from V, D, and J alleles? I mean, you were talking about association between HLA and CDR3. Did you also look at the D, J, and V alleles?
Thanks. Yeah, that is very important question. So first of all, we in addition to this association, we also tested the association between V allele versus HLA, and actually we've actually not only our research but also other previous research also suggests that there's a strong association between V allele and HLA. But we in this analysis we controlled V association, V gene association, but the association of CDR3 still remained and in very strong level. So this is most of the associations independent from V gene association, and interestingly for J gene allele, we didn't observe strong association at all. So for unknown reasons. So the fact that we observe strong association V gene but not J gene indicate that the V gene association may be mediated by CDR1 and 2 region of the V gene. Yeah, that is our hypothesis.
What about the D gene? Did you look at the D gene as well?
Oh, yeah. So actually, so D gene is included in the CDR3. So we didn't tested the D gene allele versus HLA. Yeah, that might be interesting to look at. But anyway, the D gene, we do not have many D genes. So
yeah, I think that there are 2 D genes, and only one of them has more than one allele, and I think it's only 2.
Yeah, but yeah, worthwhile to check. Yeah, thank you. Thanks for the comment.
I see that Encarnita raised her hand, so please, Encarnita.
Yes, thank you, Gore. Thank you, Hikagashi. Oh, sorry, Ishigaki.
Yeah. My name is very hard to pronounce. Yeah.
No, it's not that hard. I'm just tired. Sorry.
And this is someone who was living in Japan for several years. Okay.
I was in RIKEN,
really?
Yokohama.
Okay.
Yeah, but long time ago, before you joined.
Okay.
I have several questions, so thank you for the presentation. That was really interesting. the first one is about the tissue result that you show. So evaluation of the risk
Yeah,
score in the tissues.
yeah.
So maybe I missed, maybe you said it, but are those T cells from the tissue coming from patient that has the HLE variant associated with RA or not
Yes.
necessarily?
So basically we analyze only 6 or 7 patients, and all of the patient had at least one allele of HLA risk allele, and and also that actually that requirement is just for coming from the tetramer restriction. My collaborator used the HLA DRB10401 tetramer, so we need
Okay.
to recruit patients who have that allele.
Okay, so you did not check, did you check? Did you check for patients that don't have this particular allele, whether they also have this enrichment for those particular amino acid in the CDR3 region?
Yeah, that is actually important question, but we haven't checked that yet. And yeah, so as you know, that class 2 tetramer for
Yeah,
clinical
I know
sample
it's
is
hard.
yeah. So
I
yeah,
know.
but ideally, yeah, we should ask that question. Yeah, I totally agree.
Okay. And the second question I have, so here you focused on the beta chain.
Yes,
Uh,
yes.
so did you see anything particular? Or do you have any idea of what could happen at the alpha chain that also interacts maybe, well, of course, with the peptide, but maybe a bit more with the HLA?
Yeah, that is very intriguing question, but yeah, we couldn't test that because as you know, the adaptive just
Yeah,
provide a beta chain sequencing. But I speculate, this is just my speculation, but I speculate that alpha chain also should have some not some, but I think it's a similar level association. But the reason why I'm saying that it for V gene previous research show that for V gene HLA association alpha and beta chain, both for alpha and beta have strong association. So I speculate that for CDR3, the similar association will be should be observed. Yeah.
okay. And the last question. So when you compare the CDR3 enrichment for a given amino acid, you showed that similar patterns, in fact, in RA versus T1D.
Yeah. Yeah.
So how do you interpret this? Would that means that those amino acids provide kind of higher affinity for some antigens that would be, for instance, self-antigens, or how can that be explained?
Yeah, so that is yeah, also, yeah, that's the important question. And so basically we don't have any clear answer.
Yeah,
We need to test a lot of clinical sample to answer that question. But anyway, from genetic point of view, the HLA risk variants are partially shared between RA and T1D.
right.
So if basically this is a association result between HLA risk versus TCR. So if the HLA risk is similar, then it is highly likely that we observe similar association. So actually, this is not not so surprising result for us. It's quite reasonable. But interpretation is very difficult. RA and T1D, the pathogenic or the antigens are different. So
Totally different. Yes.
yeah.
Okay. Thank you.
Yeah. Thank you.
Okay, so thank you very much for both speakers and the audience. And we have the next seminar will be in a month, in November what? November 24. I'll put the link in the chat here, so whoever wants to attend can. And once again, thank you very much.
Thank you.
Bye-bye.